A warm welcome to all the Biotechnica subscribers. Today's topic is the previous year CSIR NET Life Science top 10 important types of question discussion. So what are the different types of questions that we get in this exam? One is memory based, in which there is no twist. It's a direct question, clear interpretation, and there are most chances that you will get it correct, provided you have a decent preparation. The other type of questions that you get here is conceptual, in which you would have to know about a particular concept in depth. These are the problems which are based on application of that concept, and you will require a detailed preparation to be able to solve such kind of questions. The other type of questions that you mostly get in Part C, they are analytical or experimental. Questions may be in the form of an experimental result from which you may be asked to derive conclusions that you must infer. And for this, immense amount of practice would be required. So is it important to get familiarized with the types of questions that come in CSIR exam? If you are attempting, then definitely yes. So why? Because if you can understand the trends and patterns of the data analysis, then it can help you predict what type of questions or which topic questions might come. It will help you prepare better. Also, it will increase the mental awareness and confidence. You will have less surprises. If you are already familiar with the type of questions that CSIR NET exam asks, then definitely the similar type of question types you will get in upcoming exam as well, so that you will be mentally prepared to answer it. And also, it will be a directed and channelized preparation. Depending on what type of questions are coming for the exam, accordingly you can prepare your topics. So you will be able to relate better. Now, what are the different types of questions that come? I have categorized into different 10 types. One are the questions which are based on correct and incorrect statements. Either you have to choose all correct or all incorrect, or maybe they will give one correct out of a set. So like that. Another will be questions on arranging and ordering. Then we have the match the following questions. We have numerical type or the formula based questions. And then we have the graph based questions. I will be telling you details about all these different types of questions and also discuss one sample question along with it so that you can understand the type. You have also diagram based or pathway related, table based questions, then experimental result analysis, or in which you have to apply the technique as I just mentioned. And another type which you get is if there is a mutation in a gene, or what will be the phenotype on it, or if you transplant some portions, transplantation based questions. These are also a type. And then you have the aptitude questions, obviously not forgetting the section A in your CSIR NET exam, right? Let's dig into the first one, which is correct or incorrect type of questions. So how to handle these kind of questions? First of all, you have to read the question properly. Make sure you know what is asked in the question, that whether they're asking for correct choices or incorrect choices. Many candidates make a mistake while reading this part. Find out read all the options and decide the most appropriate answer. So here what you can do is you can also go for the elimination method as well. Okay. So by gradual elimination of the inappropriate options, you can finally conclude to the right answer. Here is a CSIR sample question for that. This is from unit 7. The different waves of a normal cardiogram of a human subject, it is shown over here. And as you can see, they have marked different points in the graph with PQ, S and T. And they are asking which are corresponding to what is the QRS or what is the P wave or the T wave. And then you have to find out which are the incorrect statements. So this is also an example of a diagram based question. Okay, so for a diagram based question, and even this is a graph based question also, always look what is the x-axis and y-axis coordinates. Don't have any preconceived idea of what you have studied during preparation. they might change the coordinates and give you the graph. In that case, the interpretation will become different. So here the correct answer is B and D. The reason is because we know that a normal ECG tracing, it consists of waveform components, and these waveform components, they will indicate different electrical events with what happens during one heartbeat. So these different waveforms are P, Q, R, S and T and U also. Now what is the P wave over here? It is the depolarization of the atrial myocardium. So the first detection, it is associated with right and left atrial depolarization. That is why statement A over here will be correct. Second wave is the QRS. So as you can see, this is the QRS, which is depolarization of the ventricular myocardium. Here also the second wave, typically this complex has a series of 3 deflections that reflect the current associated with the right and left ventricular depolarization. Hence statement C is also correct over here. T wave, which is this one, it is normally a modest little upwards waveform which is representing ventricular repolarization. So since in B over here it is mentioned that it is arterial repolarization, that is why this B statement is wrong. Now in such correct and incorrect questions, you must be very careful with 1 letters also. Okay, so when you see repolarization, you cannot do selective reading like that. You have to be very careful about each and every word which can even go wrong, and that's why B is wrong. And even D is wrong because QT interval is measured from the beginning of the QRS to the end of the T wave. That is what is QT interval. Okay, so it is a measure of the entire ventricular action potential duration. So since in statement D it is written that QT interval indicates plateau portion of the auricular action, that's why it is wrong. So correct answer over here is B and D, which is asking for all the incorrect statements. Okay, so again this is an important topic also as well from CSIR syllabus. Next category of question is arranging and ordering questions. What you have to do over here, let me tell you a CSIR sample from here. This is regarding unit 6. Even this is an important topic, which is about the photosystem. So in photosynthetic electron transport, electrons travel through carriers which are organized in the Z scheme. Now these are the following. So what you have to do here, you have to look at the points and try to put them in sequence. Sometimes they will give the sequence in the option, or they can just give the steps. Okay, so if it is a particular technique, then they will just give you a steps. So every step you have to see and just find out which can be the first step out of that. Then you can put it in the first order. Okay, so like that, one by one, by looking at the points, you can order it yourself. And then write the arranged sequence on your rough paper, right? And then you can match with the correct option. So instead of finding the correct options, whenever this type of questions are giving, it can visually confuse you. So better is you go the other way around. Let's say these all individually, if it was given, then it would happen better you make your own order and then try to find which is matching with the sequence. Okay. And generally you can do from the starting until the end, because that is how generally we also study during our preparations, right? But be very careful of the order, whether it is increasing or decreasing, because this particular symbol, it means that this is lesser and this is more. Okay, or else you can say that this happens before and then this happens. So you have to follow the arrow, you have to know what these signs are, right? So these as I said, if you look at all the options together, if there are any wrong options, they can confuse you as well. Okay, so in this particular question, obviously option 2 is the right answer, because if anyone knows about the PS2 and PS1, so we have to know that yes, we have this P700, which is component of PS1. So from here we have this A0, A1, then ferredoxin, then FES, A, B, both are there, and then FD is there. And when we have the PS2, it starts from P680, so that's why B is correct over here. Okay, and P680, it starts with this pheophytin, then plastoquinone A, QB, then cytochrome B6F complex, then this is cytochrome B6 complex, then we have the PC, so this will be the correct, and then it goes to P700, right? So here option 2 is the right answer. Next category of questions is match the following. Now in match the following, it is very easy type of questions that you get. Now what type of questions that you will get? Either gene name or protein name or enzyme name, and what will be the function of the gene or protein or enzyme, or if it's a protein, what will be the composition, or if it's an enzyme, what cofactors can be present along with it. Here is an example in which you have to match the coenzymes, okay, and what groups they will be transferring, right? So whenever you have matched the following, try to match the easiest one which you know. For example, FAD everyone will be aware of, isn't it? Again, this is an important topic. I have tried to pick up the questions which are also from important topics as well. So you can say that these are 10 top most important questions types, okay, and also important questions. So FAD, what you have, it is, we know that it is involved in dehydrogenase enzymes, right? So the hydrogen atoms will be this. So B will be equal to 3. Now B is 3. If you see for all the B, this is the only option where B is 3. That way you can easily land up to the answer even if you don't know the other ones, okay? And try not to look at the entire table at one look. Pick one point at a time and then try to match against the available option. For example, I tried to match over here and then I tried that with which out of this will be correct. If you look at the entire table, you might get confused, okay? Now, mostly the topic of the questions are well-known data. So that is why even if you try with one or two, you will be able to get to the right answer, okay? Avoid confusing match questions. So there will be some, maybe instead of 2 columns, there will be 3 columns. Or else you will have, here you have 4 options in column 1 and 4 options in column 2. You might have some complicated tables in which you might have 2 options in column 1 and 4 options of column 2. Okay, so this can be this and this also, this can be this and this also. Like that, multiple combinations will be there. So these may look like tempting questions. Be careful of making mistake, and while you're selecting the correct option, be very careful, right? Then we have this gene protein function, or else you can get the protein composition, as I said. For example, if you see over here, integrin, cadherin, these are the cell communication, cell adhesion molecules. Again, this is a very important topic, and questions, this type of question regarding the cell adhesion molecules is also very important. Okay, so obviously over here answer will be 4, because try to select the one which you already know. Now we know that cadherin, they are calcium dependent, so that's why B will be 3. But however, you have B3 here, or here also it will be 3. But we know that select, in the word itself it has lectin over here. So D will has to be 1. Again D has 1 over here. So now it comes to immunoglobulin superfamily. Obviously, if this is immunoglobulin, that means they should have immunoglobulin-like domain. So C will be 2. That's why option will be 4 over here. What are the types you get? Organisms or properties, their characteristics or their phylogeny, such kind, or maybe the disease and their causative agents. For example, you have a disease over here, and what is the pathogen that you get, right? Here also it is a important topic, that pathogens and the plant diseases. Here the answer is 2. If you are not sure of this, then there is no way you can guess it, because powdery mildew, for example, it is caused by Erwinia amylovora. And then you have this fire blight. If you are aware of that, it is caused by this Magnoporthe oryzae. And then bacterial canker, it is done by the Pseudomonas syringae, and rice blast by this one, right? So here answer is option 2. Then you have the scientists and discovery. So for this types, of late they have started to ask the different scientists and what type of structures they have given over here. For example, Linus Pauling, we all know that he was responsible for giving the model of alpha helix. Emil Fischer, he was responsible for the lock and key model, and also the Michaelis-Menten model of enzyme kinetics. First structure of myoglobin, it was given by John Kendrew, and Christian Anfinsen, it's very popular, the renaturation experiments which gave the sequence structure relationship. Hence the correct answer over here, it will be option 2. Okay. Next type of questions are numerical type of questions, numerical type or formula based questions. What you have to know over here, obviously you need to know the different formulas which are involved. Okay. So first things first, earlier we used to do, you have to write down whatever the given things are there. Okay. Whatever is given in the question, those facts, if you write down, then you see which formula will be applicable. Very important, you must not ignore is the unit conversion. As you know, same type of units only you can do the calculations with. For example, if you have kilodalton and if you have dalton, two different units, then you cannot do the calculation like that. Either you have to convert the kilodalton into dalton or dalton into kilodalton, and then you can calculate properly. Okay, so you have to convert. When you are getting the answer, you also have to keep in mind that you get it in the required unit which they are asking. For example, if they are asking in micrometer or you have got your answer in nanometer, then you need to convert nanometer to micrometer, right? So you have to be very careful about the unit conversions. Be observant about the required unit and the powers that is there in the answer. So what does this 10 to the power minus 7 mean? In options, there might be giving some kind of plus or minus. So whether it is the plus option or minus option, be very patient and calm while calculating. Otherwise, you will be making silly mistakes. Okay. And it's better that if you are confident in such kind of questions, then you solve such questions in the beginning itself. So here is another sample from a very important concept of Hardy-Weinberg equilibrium. Red hair, it is a recessive trait in humans and it is in a randomly mating population in Hardy-Weinberg. Approximately 9% of individuals are red-haired. So what is the frequency of the heterozygotes? Here the option will be answer 3, that is 42%. Let's look at the solution. So here we have to take the dominant allele and recessive allele. The different allele frequencies we can take as P and Q. And if you know about the Hardy-Weinberg equilibrium, that if there are any two alleles for a trait in a population, then P plus Q will be is equal to 1, right? Now, genotype frequencies that you will get, the frequency of occurrence of homozygous dominant will be P square, frequency of homozygous recessive will be Q square, and frequency of the heterozygotes will be 2PQ. So that's the Hardy-Weinberg equilibrium. If there are any two alleles for a trait in a population, then all together P square plus 2PQ plus Q square will be equal to 1. Now, since red hair here is the recessive character, the proportion of population that has red hair will be homozygous recessive. That means it will be Q square. Now, Q square it is given 9%, which means it is 0.09. But since it is a square, that's why Q will be 0.3. Now you have to just put it in the equation. We know P plus Q is equal to 1. So since we have already got our Q, so P will be 1 minus 0.3, which is 0.7. Okay. So what is the frequency of heterozygotes now? You have to calculate by this heterozygotes they have asked. So 2PQ you have to apply the formula. 2 into P is 0.7 and Q is 0.3. So together you will be getting 42. You have to calculate in percentage. That's why the answer is 42%. Okay. Next type of question is questions involving graph. How to handle such questions? You have to look at the question properly. That is again when it includes graph, as I told you, just see the x-axis and y-axis. That is, read the graph properly. Correlate the text and the graph. Sometimes when such questions come, we tend to ignore the text which is written. So first see what they have asked in the question, okay, and then analyze the mechanism. You have to link the question with the mechanism by which you can solve the question. One of the question that is here, it's regarding tryptophan synthetase in E. coli cells, whether they are absence or presence of tryptophan in the media. So this is the activity in the y-axis and whether they are present or absent, okay. Now, if the two trip codons in the leader sequence of tripoperon, it is mutated to alanine, which of the following graphs will best represent activity of tryptophan synthetase in E. coli cells if they are grown in the presence or absence of tryptophan? Okay, so this is the graph which they have given, but the question is asking regarding what if they are mutated to alanine, right? Now, here the answer will be option 2. As you can see here, in the presence of tryptophan, their activity will increase little bit. So why is that? Tryptophan operon has two controls. So one is the negative control. It binds with the inactive regulator protein and makes it active, which then goes and binds with operator to stop the transcription of the operon. Then we have the attenuation control, which is mediated by the two trip codons in the leader sequence. Now, because of these two controls, when trip concentration in the cell is very high, then the operon is turned off. But when trip codon in leader sequence is switched to alanine, then the attenuation control by trip cannot work anymore. But the negative control of the operon is still there. Therefore, in the presence of tryptophan, some tryptophan will still be transcribed. Amount of it will be lesser than its expression when tryp is absent, but more than the situation in which in presence of tryptophan when both the regulations were working. Alright. Next is the questions which are based on pathways and diagrams. How to handle such kind of question? Again, you have to read the question properly and you have to read the pathway and understand it. Like what components are given in the pathway. Follow all the arrows properly because you have this arrow symbol, you have the inhibition signal, right? So you have to follow the pathway properly and you have to correlate the text and the pathway which is given. Again, try to link the question and find out what are they exactly asking. Here is a sample question. This figure shows a gene duplication event followed by a divergent event in species 1. Now you can see here this is the ancestral gene and then there is a gene duplication where it is giving a different color. If this was white color, this is giving a different color. And then they underwent this evolutionary divergence. So these two and these two, they are what? Right? So are they duplicated genes or ancestral genes or paralogs or orthologs? Now here answer will be orthologs and paralogs because orthologous genes are homologous genes in different species. That is, when a species diverges into two separate species, the copies of a single gene in the two resulting species, they will be orthologous. So A is orthologous because different color in species 1 and different color in species 2. Whereas homologous sequences are paralogous if they were separated by a gene duplication event. That is, if a gene in an organism is duplicated to occupy two different positions in the same genome, then the two copies are going to be paralogous. Paralogous genes often belong to the same species, but it is not necessary. Paralogous, it is actually of two types. The paralogous pairs that arose from a speciation event and out paralogs that arose before a speciation event. Between species out paralogs are pairs of paralogs that exist between two organisms due to duplication before speciation. So B is paralogous, okay, especially out paralog. Now you can see over here, if this is the ancestor gene and you have gene A and gene B which have got diversified, that's a speciation event which happened. Now what is in paralog? You can see that they are the pairs that will arose after the speciation event and out paralogs that will happen before a speciation event, okay. So these will be out paralog and if you have two different outcomes, you can see the A is orthologous in species 1 and here it is different color in species 2. So they will be orthologous. That's why the answer over here is option 3. Fine. The next type of questions are table based questions. For table you have to see the rows and columns very carefully. Okay, because sometimes, as I said, just like graphs, they can change the type of questions, they can change the rows and headings. Okay, it may not be the conventional table what you might have seen in your books or notes, right? Then you have to see that the description in the question, how it is matching with the table and whether they are asking like what it exactly is asking. And all data provided in the table might not be required to solve the question. So that is why try to find out what is relevant to the question that they are asking. Fine. Now this is regarding a cladogram, again an important concept from CSIR point of view. Identify the most important cladogram that can be constructed using this data matrix given below, assuming the zeros are plesiomorphic and ones are apomorphic characters. So first of all, let's understand what is this plesiomorphy. It refers to ancestral characters which are shown as 0 in the question, and apomorphy refers to derived character shown as 1. Now species having the most plesiomorphic characters, they are the one who evolved first from the common ancestor. For example, if you have here A, then you can see AB it has formed, then AC it has formed. So the plesiomorphics are the ones that have evolved from the common ancestor, right? So A over here is this plesiomorphic, right? And species having least plesiomorphic characters are the one which evolved last or more recent, right? So that is why you can see here this has evolved last. So that is why this is referring to this apomorphic character. So since over here we can see A is having all the plesiomorphic characters, that is why yes, in both of this it can be correct, A and A, they are the ones which it is derived from. and D is having the maximum apomorphic character. So that is why it should be the highest. Okay. So you can see here B is also having like this. But here C will be more ancestral than B. So that is why 1 will be not correct, and that's why 2 over here is the right answer. Right. Now next type of questions are experimental result analysis or technique application questions. So this will be very applicable in any of units you can have, but more you will see from unit 13 area. Okay. So what points to keep in mind, what you need to know: general solving approaches, thorough concept behind the question topic. Whatever is the question, you have to draw a mental image or a rough diagram of the entire process. And then if required, you can draw the flowcharts of the steps which are performed, an account of all that has been done and in what sequence. And finally, you should have a clear picture of what the technique can do or cannot do. Right? For example, this is from unit 13. Based upon phenotypic observation, it was concluded that an unknown gene which is responsible for an agronomically important trait is present in a particular plant. Now, this is an unknown gene. You have to keep it in mind. Based upon phenotypic observation, it was concluded that this gene is responsible for, in order to identify the gene. Now, in order to identify the gene, a researcher proposes to use the following strategies, right? So, which out of this? Now, an unknown gene responsible for a certain character, it can be only identified through map-based cloning and through developing of the molecular markers. So, they have asked which of the following option is most suitable for identifying the unknown genes. Okay, so this should be option B and E. Option 2 is the right answer, because with the construction of a linkage map to a genomic region linked to vernalization response in carrot, for example, the region was mapped in a linkage group with 78 molecular markers, and the flanking markers were at 0.69 and 0.79 centimorton. So this map could be used to develop molecular markers which are linked to the trait, and also in the future to start physical mapping and sequencing using a carrot BSC library. Okay. So for such kind of questions, you should not think stereotypically. Like whatever you have studied before, they can give a new type of condition. So you have to think according to that. Okay. And you have to pay attention to every detail that they are seeing. The other type of questions that you have, mutational or phenotype, what will be the mutational phenotype, or else what will be the transplantation based questions. So understand what has been done. In the question itself, they will tell. For example, you can see here what is happening after the different injections, right? Or else if you are transplanting this part to this part, then what will be. So first of all, understand from where it is taken to where it is transplanted, right? And recollect what is the role of that cell or the region or the gene, whatever is getting changed, and think about the effect of its presence or absence. Okay, what will be the effect of that gene if it is present or absent? So what you can make is, you can make columns of gene cells and functions during your preparation. That will help to retain better. And pay attention to what kind of mutation has been done. If it is mutational phenotype, like whether it is loss or gain of function, or whether it is dominant or recessive, right? And don't keep any preconceived standard mutation or transplantation result in mind. Also, you have to keep in mind which is the donor, which is the recipient, which is the host, okay, which is the donor. All these things you have to keep it in mind. So one of the sample questions over here is regarding Drosophila. Antennapedia complex in Drosophila. It contains 5 genes, which is this all 5 of them, okay, and they expressed in parasegments 1 to 5 respectively in a non-overlapping manner. So certain statements are made about antennapedia, which combination correctly describes the function of antennapedia. So if anyone would be knowing about this antennapedia, they are having this it is a homeotic selector gene which is normally turned on in the thorax area. So here you will get the expression of the antennapedia, and generally it is turned off in the head segment. So here it will be turned off. But however, if there are mutations, it can cause this antennapedia to be overexpressed in the head. Then in that case, from the head where antenna is supposed to come out, instead of antenna, leg will come out. But that will be the case when there is a overexpression. Okay. Similarly, you can have an underexpression also. So if you have this loss of mutations, recessive loss of mutation, then antennapedia won't be present in thorax also. In that case, where it is supposed to give leg in the thorax, here also you will see antenna coming. So that is why it is called as antennapedia. Now here, in the above described antennapedia mutation, the gene ectopically expresses in the head region, right? So yes, here it is ectopically expressed, means it is expressed where it is not supposed to be present. And one of the function of antennapedia is to repress genes that induce antenna development. Okay, even this is correct, because if it is expressed in the head, then it is repressing the genes that is not allowing for antenna development. Antennapedia expresses in thorax and forms a concentration gradient in posterior anterior direction. So this is not correct, because as we just saw, it is expressed in the thorax and it doesn't have any concentration gradient. And a homozygous recessive mutation will cause the leg to form into antenna even in the thoracic region. So that is why option 4 over here is the right answer. This is from unit 5, again an important topic of in the syllabus, which is regarding Drosophila. Then last concepts are aptitude questions. Now how do you handle this aptitude question? First of all, you have to manage your time wisely. Prioritize questions which are based on difficulty. Start with the easier ones to gain momentum. Use logic. You can apply logical reasoning to eliminate answer options. Sometimes basic logic can lead you to the correct answer without doing any extensive calculations. And you have to read carefully, obviously, before answering. Thoroughly read the question. Sometimes questions may require only basic reasoning or just quick mental calculations. It might just look complicated, but it might not be so much complicated. Okay. So one sample question: a long ream of paper of thickness T is rolled tightly. As the roll becomes larger, the length of the paper which is wrapped in one turn exceeds the length in the previous turn by how much? So here, if you consider this roll of paper, let the radius of the present roll be R. You can see this is R. Now, if the total circumference, length of paper covering the entire turn, we know the circumference will be given by 2 pi r. This is the formula. Now, if the paper has a thickness of T, then radius is also increasing. So now the new radius will be R plus T. That is why the new circumference will be 2 pi r plus T. Okay. So what will be the difference between the two? If you just solve it, it will be 2 pi t. So this is a very simple one. All you have to do is you have to just get a mental picture, right? So for this preparation, you can familiarize yourself with the question formats, seeing the previous year papers and different types of questions that you are getting. Practice regularly to get this aptitude concept. Okay, that's all from my end today. Thank you, everyone.